really, really happy to be joined by Juanita Mathews here. Who here knows Michael Levin's work? Okay, wonderful. Then I think you'll be quite excited by the work that Juanita does. She is in his lab and is now also discussing a microbiome challenge. So very, very excited to have you on board. Thank you so, so much for joining. And I will get your slides up in a second.
So I don't work with the microbiome, but I'm very, very interested in it. And we have done some work in our lab in collaboration with Ben Wolf at Tufts University, who does work with the microbiome. So, most people have heard this quote before, but all disease begins in the gut, and that's absolutely true. I think a lot of the problems that we see in aging originate with a leaky gut. And so you can see that gut dysbiosis basically causes a cascade of all of these different problems. You get infections, bowel disorders, cancers, autoimmune problems, definitely some neurological problems as well. And most of this can be traced to this barrier permeability that's happening in the gut and all the things that are crossing over that shouldn't be crossing over. And so there have been some really great studies that have looked at how the microbiome changes as we age. And so, of course, when we're in infancy, most of our microbiome comes from our mothers. And then, whether or not we have formula or we're breastfed, whether it was a cesarean delivery or if it was a normal delivery. So all of these things are factors that contribute to the microbiome. And then later on, what we eat really contributes to the microbiome. But what's really interesting is that the microbiome in our adulthood pretty much is very stable until about 60 years old. And then at about 60 years old, it starts to change. And what you start to see is that you lose a lot of this diversity and you start to just see amplification of, in some cases, pathogenic bacteria. And in other cases where they've looked at very older populations, like people that are getting up to 100, 105, they find that there's an amplification of bacteria that are actually beneficial. And it seems to be that these beneficial bacteria are introduced by a very varied diet, and then eventually they take over. So there's, of course, a lot that goes into how that permeability of the gut is maintained. And basically, if you lose that mucus layer and you lose that tight junctions between the cells, then you end up with all sorts of different things coming through. And most of those are protein byproducts of the bacteria that are in your gut. And those cause all of the inflammation and all the cascades from there on. And so healthy gut, we get a lot of our neurotransmitters, precursors for neurotransmitters, all of that is coming from the gut. So it's very, very important for so many things, especially the immune system and, of course, cognition. So one of the things that I was really interested in is metformin. So metformin is used in anti-aging, and they actually did a study where they put it into C. elegans, and they wanted to understand how was it actually expanding life. And they found that metformin, what it's actually doing is it's interrupting the folate cycle and the methionine cycle in the bacteria. So it's interrupting their metabolism. And so it's disrupting the normal metabolites that are being produced by the bacteria and extending life. So, the microbiome is a fascinating thing, but there's a lot of conflicting data. So some people say that, these strains are elevated. Other studies come out and find out that they're decreased, in an aging population. And so I'm really looking now at the metabolites themselves and tracking the metabolites themselves, because if the metabolites are the ones that are actually doing all of this, then that's what we should be looking at. And so my challenge is, instead of just looking at genes, instead of just looking at proteins in these blood samples that we have for aging populations, we should really be looking at these microbial metabolites, not just our own metabolites. Because they have such huge impacts on all sorts of different downstream processes. One of the big ones is sodium butyrate and indole, and this is a really great paper if you guys want to check it out, because it explains a lot of these different microbial metabolites and their targets. And so just a little bit about myself and the lab that I come from. I'm working with Michael Levin right now. I was a postdoc in his lab for about 6 years, and now I'm a staff scientist. I don't want to leave. I've got a good thing going. Mike's able to bring in quite a bit of money, and so I can just focus on science, and I get to do just really cool things. And so, if you're not familiar with bioelectricity, a lot of people are just familiar with neuronal bioelectricity or muscle bioelectricity, synapse firing or contraction. But every cell has a resting membrane potential, and that resting membrane potential is determined by the balance of negative and positive ions in and out of the cell. And so what Mike has theorized is that all cells in the body are using those resting membrane potentials to encode information about the morphology of the body itself, just like the neurons encode information about the sensory inputs that you get throughout your life, right? So what he's been working on is ways of basically actuating, changing all of these resting membrane potentials, and then seeing what effect it has on morphology, and then putting that into kind of a computational network so that we can understand and model it better. So embryonic development, remodeling, morphostasis, regeneration. We do a lot of work with regeneration. Right now we're working with mice. We were able to get partial limb regeneration in frogs past their regenerative time point. So what is the kind of cool thing that we're looking at here is that this isn't about a molecular pathway. It's basically a new top-down patterning cue. It seems to be that the membrane potential is like a master regulator. It sets the stage for all the other genetic networks that come down from that. And so what we can do, we can grow new limbs. You can see here that we have multiple limbs on that tadpole there. We can grow eyes, extra eyes. If you take a transcription factor for the eye, I believe it's PAX6, and you express it in the body, in the trunk, you cannot get eye formation. You can get it anywhere, in the head. But what we were able to do was actually induce eye formation in the tail. And not only that, the eye actually connected up with the spinal cord and was functional later on in life. So there was a frog with an eye on its butt that it could see out of. We're basically able to change these whole morphogenetic patterns by changing the membrane potential. And we do that with optogenetic channels. We do that with ion channel expression, mutated ones that are always on. And we also do it with pharmaceuticals that are ion modulating, and ionophores as well. And the other thing that we can do is normalize cancer. So that's what I've been working on. My particular project is, well, one of them is normalizing cancer. So the groundwork for that came from Brooke Chernay, who did a really, really great paper where he basically put the genes in an oncogene to create a tumor in a tadpole. And then on top of that, he co-injected a mRNA for a ion channel that would hyperpolarize the region. And what we're finding is that cancer cells and multiplying cells, embryonic stem cells, are very depolarized. And the more differentiated your cell becomes, the more hyperpolarized it becomes. And so what we wanted to do was to change that resting membrane potential, see if we could stop the proliferation. And what he found is that he was able to stop that proliferation. And so he wanted to find out what was the mechanism behind it. And this kind of goes back to these metabolites. It was a sodium butyrate that was being transported across the cell, and it was in response to the changed electrophoretic capability, so the ability of that transporter to pump in ions because of the gradient that we created by hyperpolarizing the cells. So without the sodium butyrate, you wouldn't have this effect. And so I think it's really important to understand that these metabolites are involved in everything because we've evolved with our bacterial microbiome. And so a lot of these things are dependent on one another. So this is the paper that I just published, and we actually took a bunch of different drugs that were mainly FDA approved for other conditions like epilepsy, proton pump inhibitors, and we tried out also Rogaine, minoxidil. We tried them out on cancer cells, and we came up with 13 different combos and drugs that were better than TMZ, which is the leading drug for glioblastoma. And so this type of therapy would be, you would resect the tumor because you don't want to just senesce or normalize a cancerous mass. You would take that out, and then you would do these therapies, and that would stop those cancer stem cells from proliferating and creating the cancer or having the cancer recur. And so what we saw is that this normalization made the cells senescent, and it showed differentiation factors as well, since hyperpolarization is involved in the differentiation process. And so I haven't tried metabolites in conjunction, but I think that's the next step. Another place where metabolites are also involved in some of the work, this is Frederick Lee. He was a student of Benjamin Wolfe, and so he was specifically working on microbiome research. And he found that indole actually suppressed the regenerative capability of this flatworm. And so I think there's all sorts of different effects, different pathways that these metabolites can feed into, and we definitely need more work in this area because, with metformin as an example, we could potentially, instead of trying to completely reprogram the microbiome, which is difficult, it's difficult to alter the microbiome, you could potentially just alter the metabolism of the bacteria that are existing in your microbiome and then affect those metabolites. And then you could increase the quality of life for a lot of people. So yeah, basically, we're at that point where we're on our way to a unified theory of biology that'll merge body and environment, brain, mind, genome, and microbiome, a more holistic approach, that it's not just the cells in our body, it's the other bacterial cells in our body that have a huge impact on our health.
So the specific challenge here would be what exactly?
To characterize the microbial metabolites that are in these blood samples that a lot of people are pulling, genomic information from, but to really look at these metabolites more closely and see, what the trends are over time. Also, working on therapies to alter those metabolites so that you don't get to the point where you have that impermeable gut barrier. You take care of it before it reaches that point. And then, I think that you would see a lot of good downstream effects from that.
For people over 60, what kind of therapies are currently being worked on? Would it be like a pill, would it be a shot, or would it be something to consume?
So dietary changes are big because what you eat is what feeds the certain type of bacteria that makes like sodium butyrate, for example, or indole as well. But what I am proposing basically is that we try to come up with therapies that will alter the metabolism of the bacteria directly so that we can get the right type of metabolite profile.
How do you distinguish bacterial metabolites from just whatever metabolized materials in our cells?
So we don't produce sodium butyrate and we don't produce indole. So there are definitely some that we share as far as production, but I think you could get kind of a baseline and look at, what that is.
We are interested in blood microbiome as well as metabolites, and we found it quite difficult to characterize or identify the metabolites. So do you think you have a way to categorically identify the metabolite to a concentration that you can then detect and then say that it's actually coming from the microbiome?
Yeah, I absolutely think that that's a big challenge, right? Because right now it's mass spec. That's basically what you have to do, right? And so coming up with assays, some sort of like enzymatic assay that would quickly identify particular types so that you could do a panel. I think that would be a really good idea. Just blood drops. You could do it that way. But right now, I don't think it exists, like a very high throughput way of doing this with small sample sizes. You don't want to be bleeding people just to get what you need to see this.
Thank you.